Good evening, this is Eric Michael Loyd, RBT, and also uh, have a master's in psychology, neuropsychology concentration. I wanted to read an article about longevity. This one is from Science Daily, Science News. This one is pathways that extend lifespan by 500% identified. This was posted January 8th, 2020, Mount Desert Island Biological Laboratory. Summary is that scientists have identified synergistic cellular pathways for longevity that amplify lifespan fivefold in C. elegans, a nematode worm used as a model in aging research. The increase in lifespan would be equivalent of a human living for 400 or 500 years, according to one of the scientists. Scientists at MDI Biological Laboratory, in collaboration with scientists from the Buck Institute for Research on Aging in Novato, California, and Nanjing University in China, have identified synergistic cellular pathways for longevity that amplify lifespan fivefold in C. elegans, a nematode worm used as a model in aging research. The increase in lifespan would be equivalent to a human living for 400 or 500 years, according to one of the scientists. The research draws on the discovery of two major pathways governing aging in C. elegans, which is a popular model in aging research because it shares many of its genes with humans and because its short lifespan of only 3 to 4 weeks allows scientists to quickly assess the effects of genetic and environmental interventions to extend healthy lifespan. Because these pathways are conserved, meaning that they have been passed down to humans through evolution, they have been the subject of intensive research. A number of drugs that extend healthy lifespan by altering these pathways are now under development. The discovery of the synergistic effect opens the door to even more effective anti-aging therapies. The new research uses a double mutant in which the insulin signaling IIS and TOR pathways have been genetically altered. Because alteration of the IIS pathways yields a 100% increase in lifespan and alteration of the TOR pathway yields a 30% increase. And alteration of the TOR pathway yields a 30% increase. The double mutant would be expected to live 130% longer because of those two pathways. But instead, its lifespan was amplified by 500%. Despite the discovery in C. elegans of cellular pathways that govern aging, it hasn't been clear how these pathways interact, said Herman Haller, MD, president of MDI Biological Laboratory. By helping to characterize these interactions, our scientists are paving the way for much-needed therapies to increase healthy lifespan for a rapidly aging population. The elucidation of the cellular mechanisms controlling the synergistic response is subject to a recent paper in the online journal Cell Reports entitled Translational Regulation of Non-Autonomous Mitochondrial Stress Response Promotes Longevity. The authors include Jared A. Rollins, PhD, and Eric M Rogers, PhD, of the MDI Biological Laboratory. The synergistic extension is really wild, says Rollins, who is the lead author with Jiangfen Lan, PhD of Nanjing University. The effect isn't 1 plus 1 equals 2, it's 1 plus 1 equals 5. Our findings demonstrate that nothing in nature exists in a vacuum. In order to develop the most effective anti-aging treatments, we will have to look at longevity networks rather than individual pathways. The discovery of the synergistic interaction could lead to the use of combination therapies, each affecting a different pathway, to extend healthy human lifespan in the same way that combination therapies are used to treat cancer and HIV. Pankaj Kapahi, PhD of the Buck Institute, has said. Kapahi is a corresponding author of the paper with Rogers and D. Chen, PhD of Nanjing University. The synergistic interaction may also help explain why scientists have been unable to identify a single gene responsible for the ability of some people to live extraordinary old ages free of major age-related diseases until shortly before their deaths. The paper focuses on how longevity is regulated in the mitochondria, which are the organelles in the cell responsible for energy homeostasis. Over the last decade, accumulating evidence has suggested a causative link between mitochondrial dysregulation and aging. Rollins' future research will focus on further elucidation of the role of mitochondria in aging, he said. The research was conducted at the MDI Biological Laboratory and Nanjing University, using information from double mutants developed by Kapahi. Rollins and Rogers' work was supported by the National Institute of Health, AG 056743, the Morris Scientific Discovery Fund, and the National Institute of General Medicine Sciences. P20 GM 103423 and P20 GM 104318. The source for the story: materials were provided by Mount Desert Island Biological Laboratory. And it also has a journal reference here. I'll go ahead and include that. Thank you for your time.